All right, welcome. Today, we're going to put on our detective hats and solve a real mystery, one that's happening deep inside your own brain. We're talking about fear. And not, the jump scare kind of fear from a horror movie. We're talking about the kind of deep, learned fear that really gets stuck in there. How does something totally neutral suddenly become terrifying? Well, that's exactly what we're going to unpack. Let's get into it.
So just think about this for a second. What if fear wasn't just on or off? What if it had a volume dial in your brain? A little dimmer switch you could use to turn a scary memory up, or more importantly, dial it way down. I know, it sounds like something straight out of science fiction, right? But what's so wild is that scientists think they might have just found the actual molecule that does exactly that.
So first things first in our investigation, the brain's fear center. To figure out how a fear memory gets made, you have to know where it gets made. And it turns out pretty much all the evidence points to one very specific and very important little spot.
And here it is. This is our scene of the crime, so to speak. It's a part of the brain called the central amygdala, or just the CEA for short. You can think of this tiny almond-shaped spot as the brain's fear command center. It's constantly taking in everything you see, hear, and feel. And it's its job to decide, hey, should we be afraid right now? This is the place where those fear memories are actually built and kept.
Okay, so how do you even study a fear memory in a lab? Well, scientists use a classic experiment called fear conditioning. Here's the play-by-play. First, they play a simple, neutral sound for a mouse. No big deal, the mouse doesn't react. Then they play that same sound again, but this time it's paired with a mild little shock to its foot. They do this a few times. And that's it. The mouse's brain connects the dots and basically writes a new rule. That sound is bad news. So the very next day when the mouse just hears the tone, no shock at all, it freezes. It's learned to be afraid. And just like that, a new fear memory has been made.
All right, we know the where and the how. Now it's time to zoom in and find the culprit. What's the actual molecule pulling the strings here? The researchers had a prime suspect, a molecule that seemed to be the perfect fit.
So, let me introduce you to our main suspect, a molecule called mWAKE. Now, in other parts of the brain, mWAKE has a day job, literally. It's tied to the body's internal 24-hour clock, your circadian rhythm. It helps manage the daily ebbs and flows of how active your neurons are. And here's why it was such a good suspect. Scientists noticed that mice are often better at remembering fearful things during the day. So the thinking was, maybe mWAKE is the one controlling this rhythm of fear. Makes sense, right?
So the initial hypothesis was really straightforward and honestly pretty elegant. It went like this. If mWAKE controls daily rhythms elsewhere, and fear itself seems to have a daily rhythm, then maybe, just maybe, mWAKE is the little clock inside the brain's fear center, ticking away, making fear feel stronger during the day. It was a great theory. It made perfect sense. The only problem was that it was totally wrong.
And this is the part of the story where everything gets turned upside down. The clock isn't ticking. The first piece of hard evidence came in, and it blew that neat little theory right out of the water.
Take a look at this. If mWAKE was really a clock, you'd expect to see its levels go up and down over a 24-hour cycle, right? Like a wave. But when the researchers actually measured the amount of mWAKE protein in the CAA, this is what they got. A flat line. Look at that. The levels at lights on, midday, and lights off are pretty much identical. It's not changing at all. The clock simply wasn't ticking.
So, there you have it. That really neat, simple idea that mWAKE was a 24-hour clock for fear was just wrong. But, in science, when a good theory dies, it's not the end. It's usually the beginning of a much more interesting story. The mystery just got deeper.
And of course, the scientists had to be sure, so they double-checked. They looked at the master clock proteins, the ones that are supposed to control mWAKE. And guess what? In the CTA, those weren't cycling either. But here's the crazy part. In the exact same animals, those master clocks were ticking away perfectly fine in other parts of the brain. The conclusion was pretty much inescapable. It seems like the brain's dedicated fear center doesn't have its own 24-hour clock at all.
Okay, so if it's not a clock, then what on earth is mWAKE doing in the fear center? The investigation had to pivot big time. And the researchers started thinking, maybe they were looking for the wrong kind of timer. Maybe they needed a new kind of timer.
And this led them to a brand new question, a totally different way of thinking about it. What if wake isn't a day-night clock, but an experience clock? What if its levels don't change because of the sun, but because something important happened? A timer that gets started not by the time of day, but by the act of learning itself.
And bingo. This was the breakthrough moment. Look at this chart.
It shows mWAKE levels a few hours after the experiment. In the mice who had no training, normal levels. Okay. In the mice that just got the shock but with no sound to warn them, also normal levels. But, and this is the key, in the group of mice that actually learned to connect the sound with the shock, their mWAKE levels took a nosedive. They dropped significantly. This proved it. mWAKE wasn't a clock. It was a response to learning.
So, all the pieces of the puzzle are on the table now. We have finally arrived at the solution to our mystery, and we can finally talk about fierce dimmer switch. We now know what mWAKE is really doing, and it takes us right back to that idea we started with, a volume dial for fear.
So here's how this dimmer switch actually works. It's pretty cool. Basically, mMAC's normal job is to act like a brake on your neurons, keeping them from getting too trigger happy. So when you have high levels of mGO-mWAKE, the neurons in your fear center are calm. Their excitability is low. But when that learning event happens, when the mouse learns to fear the tone, the mMAC's levels drop. That's like taking the foot off the brake. Suddenly, those neurons are primed and ready to go. Their excitability is high. It literally makes them easier to fire, which helps lock in the new memory.
I mean, listen to how the researchers put it in their paper. It sums it all up. mWAKE expression in the CA is downregulated following fear conditioning, enhancing neuronal excitation to promote fear memory formation. In other words, the very act of learning to be afraid causes this molecule to disappear for a bit. And that cranks up the volume on those neurons, helping to physically carve that new memory into the brain. The experience literally retunes the circuit.
But wait, there's one last twist in the story, and it's a really important one. You'd think, okay, if less mWAKE means more excitability, then getting rid of it completely should make for a super strong fear memory, right? Well, that's not what happened. When the scientists totally removed mWAKE, or when they just artificially cranked up the excitability all the time, it actually broke the learning process. The mice couldn't form the fear memory properly. This leads to what you could call the Goldilocks principle of fear. You don't want too little excitability, and you don't want too much. You need the amount to be just right, and that temporary drop in mWAKE is what provides that perfect window for learning.
So here's our new model, the solution to the whole mystery. mWAKE isn't a clock, it's a dynamic brake on the neurons involved in fear. Its job is to keep things stable. But when you have a significant learning experience, that brake is temporarily released, opening up a window of higher excitability just long enough to carve that new memory into your brain.
And that leaves us with a really powerful final question, doesn't it? If we're starting to understand the actual molecular dimmer switch that our brain uses to learn fear, could we one day figure out how to, well, turn it down? Could this kind of research eventually lead to new ways to help people struggling with anxiety or PTSD or traumatic memories? It's a huge question, and the science is really just beginning to scratch the surface. But it's an exciting thought. Thanks for digging into this with me today.